Hello everyone and welcome to Let's Crack NEET UG English. I'm Dr. Neela Bakore and this is our fourth lecture and we will be talking about a very special type of cell division, that is mitosis. Before we start, let's have the basic checks. Audio clear? Am I audible? Check for the audio. If everything is okay, we can get started. Okay, it sounds good. Alright, so this is the topic that we will be discussing today. In cell division, we will be talking about mitosis. In the previous lecture, we talked about one more type of division, which was a basic one, that is amitosis. Mitosis is a very special type of division. The reason is it is the most common type of division. Invariably, whenever we talk of division, it is mitotic division which we are talking about. And in this particular lecture, we are going to cover some important points related with this division. Its stages, that is karyokinesis, which is completed in 4 phases, followed by cytoplasmic division, and we will also talk about the significance of this. In these stages, there are so many important things which we need to learn. So let's get started, but before that, let's have a quick recap of the basic things which we need to know. That is, we are on the channel that is Let's Crack NEET UG English, and it is Unacademy platform. I'm Dr. Neela Bakore, your biology teacher, and you want to subscribe to the various plans which are available. The first thing which you need to do is install the app from your app store. The app is Unacademy Learning app, and as soon as you install the app, you get access to all these things. That is your daily live classes, for your syllabus, regular tests, which is the most important part, because that only helps us whether we have understood the concept clearly or not. And you would have unlimited access to the live lectures which are conducted by the top educators of country. Here we have a list of few top educators who are training students preparing for NEET UG for years. And this is the basic plan, and let us not forget that if you want to choose any plan according to your choice, if you want to take this advantage of 10% discount, you will have to use my code so that you are eligible for that. And I told you, and I'm rather telling you every time, that the plan which you should be selecting should be of a longer duration, because one, you get a rapport developed with the teacher, you start understanding the teacher, and it is better if you go with one teacher or 2 teachers so that that flow is maintained. And we need a long-term preparation for this competitive exam. So choose the plan accordingly, like you can go for 1-year plan, you can go for 2-year plan, depending upon which grade you are in right now. Coming back to our topic, and this is mitotic division or mitosis. Mitosis is known as equational cell division. Important. Equational because the 2 daughter cells formed have the same amount of genetic material, the same number of chromosomes as that of the parent cell. If this is a cell with 46 chromosomes, after division, after mitotic division, the 2 daughter cells are going to have 46 and 46 chromosomes. So this is the reason it is called equational division. Let us talk about the discovery part. In plant cells, it was discovered by Strasburger, and in animal cell, it was discovered by Fleming. 2 scientists, important. There are very few scientists, maybe 10-20 scientists whose names are very important, and in the beginning, when we just started our sessions, I told you that in biology, there is only 5-7% things which are to be remembered or mugged up. Everything else has to be understood. And in those remembering things or mugging up things are one, the names of the scientists and their discoveries, and second is some years or something. Other than that, everything has to be understood. So same thing is applicable in this particular division also, that we have to remember just the scientists and then everything is to be understood. It takes place, this division takes place in somatic cell. Now this information is according to our NCERT. NCERT says that mitotic division takes place in somatic cells. But when we talk of meiotic division or when we talk of gamete formation, in gamete formation, that is spermatogenesis or oogenesis, there is one step which is multiplication. And in that multiplication, the division which is taking place is mitotic. But if a question is asked in our NEET exam that mitosis takes place in, so our answer is going to be somatic cells. It actually is that somatic cells divide only by mitosis. But remember, we have to stick to the statement which is given in our NCERT. 2 steps: karyokinesis, cytokinesis. Karyokinesis is nuclear division and cytokinesis is cytoplasmic division. Alright? Karyokinesis is completed in 4 phases. We can write it here if you want. Okay. It is completed in prophase, metaphase, anaphase and telophase. So these are the 4 phases in which this will be completed. We are talking about the first phase, that is prophase. Maximum changes are going to take place during this phase. And what we will do is, we will start with an interphase cell. A cell which has completed interphase. We have talked of cell cycle, we have understood cell cycle. So interphase is done, that means G1, S, G2 is done, and now the cell is entering into M phase. In M phase, first was karyokinesis. In karyokinesis, the first step is prophase. And we are talking of animal cell. And then towards the end, we will see how is it different in plant cell. So if this is our animal cell, and we will take all the basic numbers because we want to know or understand how come the 2 daughter cells get the same number of chromosomes as that of the parent cell. So this is an interphase cell. It is an animal cell, so animal cells have centriole and the nucleus is large. It has a large nucleus. Inside the nucleus, these are chromatin fibers. Fibers are visible, and we take 2N cell. So for our convenience, we are taking only 4 chromosomes. So here we have taken 2N is equal to 4. So when the 2 daughter cells are formed, each one should have 4. Then only this division is going to be equational division. This is for our convenience. We have drawn few chromosomes or chromatin fibers. One thing which we have to remember is, in most of the stages of cell division, we do not label except for very few. So here I am not going to label anything. We do not draw any organelle because our main focus is on the nucleus. And because it is an animal cell, we have a centrosome here and 2 centrioles inside. Now this cell enters prophase. Prophase is the longest phase of karyokinesis and mitosis. And in this, there are stages, like there is an early stage, then there is a mid stage, then there is late stage. There is no demarcation that, okay, the early ends, the mid starts. They can be overlapping, but there are changes which are going to be very significant in early and late. So what we'll do is we'll draw 2 stages. Let us say we are drawing the early stage. So this is our cell and this is early prophase. What happens in early prophase? In early prophase, the nuclear membrane starts to dissociate. Is there a difference between the 2 nuclear membranes which we have drawn? One in the interphase, another in the early prophase. In interphase, the nuclear membrane is very prominent, distinctly visible, and we have drawn it like that because nuclear membrane is porous. So we normally do not draw solid nuclear membrane. And in early prophase, we see that the nuclear membrane is dissociating. Inside there is a nucleolus also. Here you would see that the nucleolus is also dissociating. We'll write down all those changes. So nuclear membrane starts to dissociate. How come it dissociates? Because in G2, if you remember, G2 phase of interphase, there were enzymes which were synthesized. One category of enzyme was lytic enzymes, which were meant to dissolve this membrane, and that is how this membrane is getting dissociated. Not only this, the membrane of the organelle will also dissociate. So here, nuclear membrane, nucleolus, organelle membrane start to dissociate. Let us come to the centriole. Centriole splits in S phase. In S phase, there were 3 things which were happening. DNA replicates, centriole splits, and histone proteins were synthesized. So the centriole has already undergone this splitting. So now what happens is, one centriole remains at one end or one pole, and the other centriole piece starts to move towards the opposite pole. It starts to move, it has not reached the opposite pole. So split centrioles start to move to opposite poles. So this is going to be one pole, this is going to be the opposite pole. What is happening to the chromatin fibers? Chromatin fibers are undergoing condensation, dehydration, and now they will be visible as slightly thicker structures. Not very thick, but the condensation has begun. So what one more change we can write here? Chromatin fibers start to condense. This is what is happening in early prophase. Then there would be mid-prophase. In mid-prophase, furthermore condensation will take place. The centriole which has started moving will reach closer to the pole. More and more dissociation will take place. So the same change is going to continue. So what we'll do is, now we will straight away draw late prophase. What has happened in late prophase? The nuclear membrane has completely dissociated. Nucleolus is completely dissociated. So, nuclear membrane, nucleolus, organelle membrane completely dissociate. So the entire space is now available for the chromosomes. The two centrioles have reached two poles. And one very important thing starts here. Some fibers start to appear. Spindle fibers and aster rays appear. Appear means tubulin protein was already synthesized in G2 phase. Now only polymerization of that tubulin. Tubulin was formed in the form of a monomer. Now these monomers assemble and that is how you get a thread-like structure. That is what is fiber. So polymerization is going to take place. So let us draw these changes which we have written so far. First, the centriole. So this is the centriole, here is a centriole. The two centrioles have reached two poles. Now around these centrioles, small fibers appear. These small fibers, they are called the aster rays. Smaller ones. And then there are some longer fibers. The longer fibers are of two types. One is going to run like this. The other one runs like this. I'm going to draw the chromatin which is changing into chromosome first, and then we will come to the other fibers. Let us draw these chromosomes. They have become thick, and because the entire space is available for these chromosomes, they are everywhere. Okay, so now I'm going to draw this chromosome for you separately. And here is this kinetochore. Okay, how much is the genetic material with this chromosome? It has completed interphase, that is G1, S and G2. That means each chromosome has double the amount of DNA, or in other words you can say that each chromosome has two sister chromatids. So this chromosome is actually like this, every chromosome. And now let us come back to those spindle fibers. So there is one fiber which is attached here, one here. Okay, so we find two types of spindle fibers. What we will do is, we will draw this diagram even bigger separately also to understand the detailed structure. But here itself, we can see two types of fibers. What about this one? It runs from one pole to the other pole. It is not connected to any chromosome. So this fiber is called continuous fiber, or It is also known as interpolar fiber. What about this fiber? It starts from one pole, but it is connected to the chromosome. So it is called chromosomal fiber, or it is known as discontinuous fiber. So there are two types of spindle fibers. And what about these tiny ones? These tiny ones are called the aster rays. And this central thing which we have drawn is the centriole. So one centriole along with its aster rays, that means if you're talking about this structure, what all things are there in this structure? There is one centriole and the smaller fibers which were around it. And this structure is known as aster. So spindle fibers have appeared, aster rays have appeared, and two asters are formed. What is there in an aster? An aster has a centriole and its aster rays. The spindle fibers are of two types, continuous and discontinuous, depending upon whether they are attached to the chromosome or not. So, as I said, I am going to make that diagram bigger so that it is easier for us to understand. Let us choose the same color so that there is no confusion. Okay, so this is our big cell which is in late prophase. In this cell there are now chromosomes. Each chromosome is with two sister chromatids, and the chromosomes can be anywhere because the entire space is available for the chromosomes. All the membranes and organelle, they have dissociated, their membranes have dissociated, and so the entire space is available for these chromosomes. These are the two centrioles, and let us draw their aster fibers. So these are the smaller fibers, the aster rays. And now let us come to the spindle fibers. And we can see two types of spindle fibers. We are going to label it here also. These are the continuous spindle fibers, and they are also known as interpolar fibers. And the ones which are attached to a chromosome, they are called discontinuous spindle fibers or chromosomal spindle fibers. This structure which we have drawn in blue is the centriole. These tiny fibers which we have drawn are aster rays, and these two things together form the aster. How many asters are formed? One aster at one pole, other aster at other pole. So I am writing an important statement here again. In animal cells, mitosis is amphiastral. Amphi here is for two and astral is for asters. But the condition is it is an animal cell, because for an aster to form, centriole has to be there, and in plant cell there is no centriole. So this is one. Second, in plant cells, mitosis is, what should we call it? Will the aster be formed? For an aster to form, centriole is required, and plant cells do not have centriole. So as there is no centriole, there is no aster formation. So the mitotic division will be termed as anastral. So this is again a very, very important thing. Amphiastral in case of animal cell, anastral in case of plant cell. Very important. Questions are asked in competitive exams on such type of things also. So now what has happened till the prophase is completed? Let us go back. We started with interphase. Interphase cell has completed G1, S and G2, and now it is ready to get into M phase. M is multiplication, and here the multiplication or the division that we are talking of is mitosis. First phase or first part is karyokinesis, and in karyokinesis the first step is prophase. In prophase there are three stages: early stage, mid stage and late stage. In early stage, the membranes start to dissociate. Nuclear membrane, organellar membranes and even nucleolus, so that the complete space can be made available for the chromosomes, so that proper distribution can take place. Chromatin fibers, they start condensation, so they become a little thicker. Centriole, which has undergone splitting in S phase of interphase, now moves to opposite pole. This process begins. Mid prophase, the same things continue, and by the late prophase, one more structure appears, and that is the spindle fibers. Now spindle fibers, why are they called spindle fibers? Because the arrangement is in the shape of spindle. This is the spindle shape. 1 point, 2 point, narrower at the poles and wider in the middle. This is the spindle shape. So because these fibers, they are arranged in the spindle shape, they are called spindle fibers. And these spindle fibers, they are made up of tubulin protein. And this tubulin protein was synthesized in G2. So smaller fibers appear, which are called aster rays. Longer fibers appear, which are called spindle fibers. The spindle fibers are of two types. One, which are attached to the chromosome, they are called chromosomal fibers, and because the chromosome breaks this fiber, they are called discontinuous. And there are some fibers which run from one pole to another pole without connecting to any chromosome. They are called continuous fibers or interpolar fibers. To understand this structure in detail, we made the late prophase bigger. Here we are seeing aster. What is an aster made up of? An aster has centriole and aster rays. So this becomes the aster. As in animal cell, centrioles are present, and the two centrioles move to opposite poles, there are two asters formed, and that is why the division in animal, mitotic division in animal cell is called amphiastral. In plant cell, there is no centriole, so no aster formation, and that is why the division is known as anastral. All these things take place in prophase. So it is a long phase. Many changes are taking place here. These are all the changes which are taking place. We have written down those changes in points so that it is easier for us to recap. It is the longest phase, We talked about it. During early prophase, what happens? All these membranes start to dissociate, okay? The split centriole, they also start to move to opposite poles. Mid-prophase, chromatin fibers, they start to condense, chromosome-like structures start to appear. Chromosome is having two sister chromatins. Important point, very, very important point. One chromosome, because it has undergone S phase, it has double DNA, and these two are known as the sister chromatids. So this we have to keep in our head, that every chromosome is bringing double DNA. During late prophase, aster rays, spindle fibers, their assembly takes place, they appear. Mitotic apparatus is fully formed. What is this mitotic apparatus? Both the asters, spindle fibers collectively form mitotic apparatus. Very simple. What we made was a cell, or let us not draw the cell. Let me just start with the centriole. Let us choose the same color. So these are the two centrioles. Let us draw the aster fibers around them and the spindles also, spindle fibers also. Some continuous, some discontinuous, and to the discontinuous ones are attached the chromosome. So let us make one chromosome here, one here, what have we drawn? We have drawn aster, we have drawn another aster, we have drawn chromosomal spindle fiber, and we have drawn the interpolar fiber also. We have not drawn the cell membrane. So if you remove the cell membrane and you draw only two asters and spindle fiber, then that is known as collectively mitotic apparatus. We have drawn the two types of spindle fibers also, continuous and discontinuous. Now, the next stage is metaphase. And in metaphase, all the chromosomes, they will come in the midline. If this is a cell, this is one pole, this is one pole, so this is called the equatorial plane. Like on the globe, we have north and south pole, and then there is an imaginary line which is the equator. Similarly, in a cell, the two ends where the asters are, these are the poles. And the midline, there is no line, it is just an imaginary area. That is called the equatorial plane. All the chromosomes will come here at the midline. How are they going to come here? Let us take this chromosome and say this is the midline where they have to come. How will this chromosome, this one we are talking of, how will it reach here? It has to be brought. So between prophase and between metaphase, all these movements of chromosomes will take place due to contractions of these spindle fibers. And this intermediate stage is also there. And this intermediate stage either is called intermediate or a transition stage. During this stage, what happens? There is differential contractions of spindle fibers of chromosomal spindle fibers. And because of this, now the chromosomes will come to the equatorial plane. Again, let us draw that structure quickly. This is the centriole, this is the second one. All the basic things must be there. The aster rays and the spindle fibers. This is the chromosomal one. The place where I have not drawn this spindle is the place where the chromosome is going to come. And these are the continuous fibers. Let us place the chromosomes now. Okay. Now, let us draw that imaginary line also. This is the imaginary line which is the equatorial plane. So if we are talking about this chromosome, how will it come here? It is attached to the chromosomal spindle fiber. So now there is a part of chromosomal fiber which is from one pole. This part is not going to contract. This part, this spindle fiber contracts. So what will happen? This chromosome will be pulled down. Same thing is going to happen here. This chromosome will be pulled down because this fiber is going to contract. In this case, this part of the fiber will contract and this chromosome will move. Here also this will contract and the chromosome will come. And that is why we have used the term differential contraction. All the fibers are not going to contract equally. Some contract towards one pole, some contract towards the other pole, someone would be more, some fiber would be less. But this is an intermediate stage. And as I told you in the beginning, there is no clear-cut demarcation that prophase ends, metaphase starts. So there is a transition phase between prophase and metaphase. And that transition phase is this intermediate phase. Clear so far? Now, after this, the next phase starts, that is metaphase. And if you have carefully seen everything, you must have noticed that we made the labels, or we drew the labels only in one stage. That is when we were trying to show the fibers and all those things, because this is something new. We do not label nucleus, no organelle, nothing, except for fibers and aster and those things. This is our cell. Which is in metaphase now. Let us draw all the structures which are as it is. The two centrioles at two poles. The aster rays, which are the shorter fibers. These are the shorter fibers, aster rays. Here also aster rays, and the spindle fibers. But now you would see that all the chromosomes, they have come or they have reached the equatorial plane. And we have to draw both the types of fibers, the chromosomal and the interpolar fibers. Let us come to the chromosome. So this is one chromosome, this is the other chromosome, the third and the fourth. Spindle fibers are attached to the kinetochore. So if this is the chromosome and here is that kinetochore, the spindle fibers are attached to it here. So to one chromosome, the spindle fibers which are attached, one is coming from one pole, other is coming from the other pole. So if you see, this fiber is attached to this chromosome, same, and another fiber from the other pole is also attached to the same chromosome, or we can say the centromere of the same chromosome. And don't forget, this chromosome has double DNA. It has sister chromatid, its original and the copy DNA. So what is happening in metaphase is that all the chromosomes, they align on the equatorial plane, and every chromosome which is there at the equatorial plane is actually having two sister chromatids, or in other words we can say that it is having double the amount of DNA. Let us see what are the changes that we have discussed. The same changes, do we have those here? Chromosomes arrange at the equatorial plane. This equatorial plane is also known as metaphase plate. You can call it equatorial plane, you can call it metaphase plate, anything. Centromere of each chromosome is attached to two chromosomal fibers, right? Let us go back to this. See, each chromosome is attached to two chromosomal fiber, one coming from each pole. And an important point, chromosomes are shortest at metaphase, and because of which metaphase is the best phase to study structure of chromosomes, because the chromosomes are shortest, maximum condensation has taken place, so they are short. So because they are short, they can be studied in a best possible manner. This is the second phase. Now let us come to anaphase. Our cell has entered anaphase. So first we will draw the cell and we will see what is going to happen in anaphase. Centrioles, the basic structures that we have to draw. So two centrioles are here. Let us draw the aster rays. All these structures must be drawn, but we are not going to label it after we have done it in prophase, late prophase. And now these fibers, and this is the continuous one. The chromosomal fibers have contracted. Let us come to the chromosome now. Okay, we will draw it outside first and then we will place that chromosome inside. This was the chromosome with its kinetochore. The spindle fibers are attached here. Now when these spindle fibers, chromosomal spindle fibers contract, the centromere splits. So because of this, now you would find that it is going to split like this. So one part goes towards one pole, the other chromosome goes towards other pole. Centromere has undergone splitting. So this is one, there were 4 chromosomes. each one having double DNA or each one having sister chromatid. So now when splitting takes place, 4 chromosomes with the normal amount of genetic material go towards one pole and 4 go towards the other pole. In this case, we will add one more thing. Between the split centromeres, a fiber appears. So now there is one more fiber which is there. We called this fiber, which goes from one pole to the other pole without getting attached to any chromosome, as interpolar fiber. It was also called continuous fiber. Then we have this one which is attached to the chromosome. It is called chromosomal fiber or discontinuous one. And now there is a third fiber which appears, and this is known as interzonal fiber. Interzonal fiber appears only between split centromeres. So if centromeres split, then only we will see interzonal fiber. Otherwise, interzonal fiber is not seen. So in which stage do we see interzonal fiber? Is anaphase of mitosis. This statement you have to remember, because when we come to the next division, that is meiosis, I will be asking you this question again, and there the process is even more complicated. We will have anaphase 1, anaphase 2, and we will see in which one is interzonal fiber formed. But for the time being, what we have to remember is interzonal fibers appear only between split centromere. Otherwise, you would not see this interzonal fiber. So what has happened in anaphase? Again, in anaphase we can have early, mid and late. In early anaphase, the chromosomes have just started to separate. The short interzonal fiber is formed. By the late anaphase, the chromosomes will reach towards the pole, and there will be a longer and longer interzonal fiber. Now, what are those things which are moving these chromosomes apart? Or what are those forces which are responsible for taking these two chromosomes apart? One, this chromosomal fiber is contracting. So first is contraction of chromosomal spindle fiber. Second, this interzonal fiber is going to stretch. It's like we have two threads and there is a fiber which appears in the middle. Suppose this fiber stretches, these two threads are going to move apart. Stretching of interzonal fibers. And third thing, the very important thing is ATP. And that is why in G2 phase, lots and lots of ATP molecules are to be synthesized and accumulated, because to carry one chromosome from the equatorial plane to the pole, about 30 ATPs are required. 30 ATPs are required to move one chromosome from equatorial plane to pole. So here in this particular cell, there are 4 chromosomes which are moving towards one pole and 4 chromosomes which are moving towards opposite pole or the other pole. So how many ATPs would be required? 4 into 30 to move towards one pole and 4 into 30 to move into the other pole. And 4 chromosomes we have taken only for our understanding, so that if there are less chromosomes, all the stages and everything will be clearly visible to us. Think about our cell. We have 46 chromosomes, and if that 46 chromosome containing cell divides by mitosis, so 46 chromosomes will go towards one pole, 46 will come towards the other pole. Into 30, this much ATP will be required when one cell divides only by one mitotic division. So amount of ATP required is very high. And do you remember in the previous lecture we talked about difference between amitosis and mitosis? We said amitosis is less expensive because you have to just break the nucleus, and mitosis and meiosis are more expensive. Now you see how much ATP is required. So that is why we write that these two divisions are expensive divisions. So this is anaphase. Now let us see whether we have all the changes written down here or not. It is the shortest phase. The duration is short. The chromosomes are thickest during this. They are shortest during metaphase. They are thickest here. So when they are shortest, it is the best stage to study them. Chromosomes split by splitting of centromere. This is an important point. Interzonal fibers appear between the split chromosomes. Important point. The forces which help in movement are 3. Contraction of chromosomal, stretching of interzonal, and ATP. Important. Very, very, very important points. Now, if you have studied the different types of chromosomes, you would know there are metacentric, submetacentric, acrocentric, and telocentric. If you have not done that, that's fine. I'm just going to give you a basic idea because that is associated with this phase. If the length of 2 arms chromatids is same, or if the centromere is centrally placed. This is one possibility. The second is one arm is shorter, the other arm is longer. Second type of chromosome. The third, one arm is very short, other arm is very long. And fourth, centromere and there is only one arm. This chromosome is called metacentric. One short, one long arm is called submetacentric. This one is called acrocentric. And with only one arm is called telocentric. Now, if these different types of chromosomes are being pulled by the spindle fibers, so how will those spindle fibers or how will these chromosomes look like? Let me draw 4 spindle fibers, 1, 2, 3 and 4. Suppose the metacentric chromosome is being pulled. So this metacentric chromosome is going to look like this, 2 arms. The structure to which the spindle fiber is attached will be pulled first, and the free structures, that is the arms, will be pulled later. If you're talking about submetacentric, then one arm is shorter, the other arm is longer. If you're talking about acrocentric, one arm is very short, other arm is very long. And if you're talking about telocentric, then the arm is like this. Don't you think they resemble some English alphabets? This resembles K, or it can be written as only this. This resembles L, this resembles J, and this resembles I. So, these shapes of chromosomes will be visible only during anaphase, when the spindle fibers are actually pulling them. So this is only if you know these types of chromosomes. The last part of karyokinesis, that is telophase. Telophase is actually reverse of prophase. So this is our cell, and when telophase starts, you would start seeing that there is a constriction which appears in the plasma membrane. Okay, a constriction appears. Let us draw the centriole. What happened in prophase? The nuclear membrane dissociated, nucleolus dissociated, organelle membrane dissociated, fibers appeared. Chromatin fiber condensed to become thick. Everything is going to happen in a reverse manner. Nuclear membrane will reassemble and it will be visible now. So now nuclear membrane appears, it becomes visible. Enzymes are required which are synthesized in G2 phase. Chromosomes, they decondense and you will start seeing chromatin fibers. How many would be there in one side? How many chromosomes are moving towards one pole? 4. So when these chromosomes decondense, we would see 4 thread-like structures. 1, 2, 3, 4. Here also 1, 2, 3 and 4. Spindle fibers dissociate and disappear. Aster rays dissociate. Nucleolus assembly takes place and it reappears. Everything reverse of prophase. Let us see. Changes opposite to prophase. Nuclear membrane, nucleolus, organelle reassemble and now they start appearing, they are visible. Chromosomes decondense and start to appear like chromatin fibers. Aster rays, spindle fibers also dissociate. Everything just reverse of prophase. Telophase is done, nucleus has divided. We started with one nucleus and now there are 2 nuclei, each having the same number of chromosomes as that of the parent cell. Nuclear division is complete. Now the cell divides its cytoplasm, that is cytokinesis. So now what happens here is, in the first stage, this cytoplasmic constriction which is formed, it becomes deeper. Next stage you will see that it becomes even deeper. Next stage, the cytoplasmic division is complete and you would see 2 daughter cells. Each cell is having its own nucleus and each nucleus is having 1, 2, 3, 4 chromatin fibers. And because it is an animal cell, we should not forget making the centriole. So here is the centriole, here is, every cell is having its own centriole. And these are the 2 daughter cells. In animal cell, cytokinesis takes place by constriction in plasma membrane. and it starts from periphery and goes towards the center. But cytokinesis is slightly different in plant cell because plant cells have a rigid cell wall, and if plasma membrane has to show folding, like in this case there is an infolding, it should be exposed. If it is covered, the plasma membrane cannot show folding. So because of the rigid cell wall in plant cells, plasma membrane cannot show folding. So cytokinesis in plant cells is slightly different. Let us see how it happens in case of plant cell. So this was our cell, no constriction, but two nuclei are formed, and it is a plant cell, so no centriole. Now, some fragments appear in the middle. These are called phragmoplasts. What are phragmoplasts? These are pieces of endoplasmic reticulum and Golgi body. And what do? They secrete middle lamella. So now what happens is, in the middle there is middle lamella secreted, and this is going to grow towards the periphery. So this plate is also going to grow towards periphery. Let us make the cell. This is the nucleus here, this is the nucleus here. Now, the cytoplasm of each cell deposits cellulosic cell wall on either side of this middle lamella. So this cell secretes its cellulosic cell wall, so cell wall gets deposited here. This cytoplasm also secretes, so that also gets deposited here. So a cell wall is formed, and it is going to grow on all the sides. A stage will come when the cell gets completely divided into two daughter cells. It has its nucleus, this also has its nucleus, and the wall which is formed in the middle, this is the cell wall. Middle lamella is secreted by the phragmoplast, and this cellulose is secreted by cytoplasm. And these two cells which are formed, they are the two daughter cells. Okay, so this is how the cell division by mitosis is completed. What is the significance of this division? Mitosis actually helps in keeping the number of chromosome constant, because the two daughter cells which are formed, both of them have the same number of chromosomes as that of the parent cell. So the number of chromosome is going to remain constant. It helps in growth. The zygote is formed and then the division is mitotic. That is how we grow. If some tissue has to be repaired, suppose there is a cut in the skin and new skin cells are to be formed, the division which takes place is mitotic. So repair, regeneration, everything which takes place is by mitotic division. And in lower organisms like amoeba, this is the main method of reproduction. They do not show sexual reproduction. So lower organisms reproduce asexually, and the division which takes place is mitotic division. So this is our first main type of division, that is mitosis. So I hope you have understood all the stages. Revise all the stages, go through all the diagrams and the changes which take place. And again, coming back to the same, if you want to go for a particular plan of subscription, don't forget to use my code so that you get that 10% discount. So this is for this lecture. I will see you in the next lecture with the next type of division. Till then, keep revising, stay safe, and I'll see you later. Bye-bye.